Welcome to the Cancer Care Breakout Session for the Individualizing Medicine Conference. We're very glad to have you here today. We have a very distinguished panel of speakers here, and I will introduce each of them before they come up and speak. Each speaker will have about a 20-minute talk, and following at the completion of all the talks, we will actually have a discussion session. And with regard to that, I just wanted to bring up a couple housekeeping issues. If you notice on your smartphone or your iPad or whatever you've got there that the IM website is not up in this room, this is actually a theater, and they want the focus on the arts, so they block the wireless. But actually, we've arranged for actually some wireless in here in this room through Mayo Guest. So if you actually go to your browser and type on Mayo Guest, you should be able to log in through there. You have to accept that, and then ultimately go to the IM website, and that will allow you then to submit questions, and then we'll deal with those questions at the end. So without further ado, I'd like to welcome Dr. Fonseca up to the platform. I worked with Dr. Fonseca a few years ago, and just I have to say, I guess I'm going to introduce myself. My name is Dr. Getz. I'm a professor of oncology and associate professor of pharmacology, and it's the G-O-E-T-Z Getz, so Dr. Fonseca is the Getz family professor of medicine. He's actually the chair of medicine at the Mayo Clinic in Arizona. I'm not going to go through all of his accomplishments, but you can see that Dr. Fonseca has really been a leader as it relates to research in terms of genomic and clonal cell issues as it relates to myeloma. He's had R01, P01, as well as SPORE funding, and we welcome you, and thank you.
Thank you very much, Matt. It's a pleasure to be here. I've never noticed that, but actually that's a great point. I lost the O when I went down to Arizona. So thank you for coming here. We're going to chat a little bit about cancer. I'll tell you, my talk is largely conceptual, and then there's some anecdotes. And as I was thinking about the future, I think if I was to do this a year or 2 years from now, I suspect there would be more anecdotes. And that's going to be one of the challenges. How do we put it all together? Now, one of the favorite topics, you've all seen this painting, but we've all practiced personalized medicine for centuries. This was personalized. What we really are getting to is precision. How do we actually do the individualized approach to the specific patient with their host biology and the tumor biology? Point number one with this picture. Point number two, everyone loves this picture. There's a lot of compassion. You see a mother suffering in the background. You see the physician trying to help this patient. But it tells out he has almost no tools. He might recommend a bleeding or perhaps something of that nature. And we really have the power now to change what we're doing in medicine. And that's the excitement. We can go beyond just the empathy, the sympathy, and the compassion that you actually see in this picture. Everyone's shown pictures like this before. I just wanted to show it once more. It seems like there's a stark difference in the mood between Dr. Collins and Craig. So I don't know exactly why that was. I assume there's probably a back political story. When you're a cancer patient and you're dealing with the possibility of genomic testing, the cancer patients have one question, and only one question that matters, and that is, will this help me? They kind of assume everything else is solved. They really don't care about everything else as their priority. Their priority is, will this type of testing help me? Will this type of testing cure me? Can it make me live longer? Much like when we get on a plane. Can I get to the place I'm going? We don't really care about the fluid and the brakes. We kind of assume that's there. We just want to know that we're going to get where our plane is taking us. And that is, in my mind, how I look at clinomics or clinical genomics. And I think as we talk about all the questions, we talk about policy, we talk about everything else, I think the primary discussion is between those in the front line, the oncologists and other healthcare providers, and patients and their families as teams that provide them advice. Today I was tasked to provide a perspective on challenges, and I'll disclose by saying I'm very optimistic. I'm sort of one of the most strong supporters of this technology, but at the same time, I do recognize some of the challenges that we have for genomic testing. And you've heard about them throughout the session, the administrative and technical ones, such as lab testing, quality, cost, the turnaround time, whether it's available or not, so the medical aspects of testing, how do we need a biopsy, how to process a tissue, when to test, how many tests, the ethical considerations of dealing with the proposal, as well as with the results. And then the most important ones, once you go through all of that, again, you get back to the primary question, will this help me, are the practical ones. Can we find a target? Can we find a drug? Will the drug work? Can I afford it? Or can we afford it? And how fast can this be done? Because that's really at the forefront of what the patient is thinking when we see them in the clinic. Now, a few years back, I developed this slide where we tried to think about how we would integrate genomics into the clinic. And I'm just going to build it quickly to say that when you look at the different tumor types, genomics can position itself at different points in the course of the disease for a person. More so a year or two ago, people had this concern, well, what about the standard of care? Will genomics disrupt the standard of care? Well, there's a bit of news that's very important. Sometimes the standard of care is terrible. The standard of care for some of the tumors that we treat is very poor. For some tumors, we can do better. So we can cure people with breast cancer, adjuvant therapy for breast cancer. We can even cure, for instance, micrometastatic disease with colon cancer, the disease I deal with, myeloma. We have very good options. So I think it probably would not be a very good idea to, at this point, do genomics up front so then I can change what I can do. I have good tools. I'm going to save that for down the line. But you've heard stories at this meeting, unknown primaries, primary refractory tumors, that venture out to say things such as advanced pancreatic cancer. If you're not responding to the first line of therapy, you're in big trouble. So we need to think about where along that spectrum of the life of the disease for the patient we want to position genomics. Now, one of the themes that has been managed at this conference is how do we classify diseases? And this is actually where it starts, that we thought about cancers according to the organ of origin. And this goes back to the mid-1800s, where people said, well, we need to classify the diseases. Let's look at below and above the diaphragm, the lungs, the heart, etc. And that served a good purpose for 100 years. But as you have heard throughout this meeting, there is a possibility that we'll look at cancers by genetic similarities as opposed to the tissue of origin. And if you do that, this is an example of a disease I don't deal with, lung cancer. When I was in training, we had this idea of the non-small cell and the small cell lung cancer. Then we learned about the mutations, KRAS, etc. EGFR came along. And this is kind of a more current perspective of how people are breaking lung cancer. The point being here, we understand it better. And guess what? Some of the most interesting findings are actually occurring in non-smokers. They have a different mutation rate. So it's sort of a different disease. We've already accepted the use of genetics for clinical decision-making. This is something that many of you are familiar with, the case of breast cancer, where you don't really actually need to see those curves. You just know that through genetics, you can propose to certain patients whether they get chemotherapy or not. And I'll suggest to you that for a young woman, the idea of not losing her hair is a huge benefit. It's of immense value to that particular patient. So I would say that has been tremendous progress. Put aside all the potential toxicities and complications that patients can have. But one of the things that worries me the most, I'm going to show you a couple of examples for this, is this idea of genomic evolution and the clonal and the subclonal nature of cancer. And I venture to say in the next 10 years, probably the most important prognostic marker for most of the cancers we see, and we're seeing signals for this, is how unstable is that tumor? Because tumors that are unstable have a propensity to find loopholes around some of the roadblocks that we're trying to put with individualized medicine, with small molecules, etc. This is a whole genome view of different forms of breast cancer, and just by looking at it, I'll tell you going up is a gain, going down is a deletion, good prognosis. You can imagine patients in the center, triple-negative breast cancer, the most aggressive variant of the disease. And as much as the specific markers matter, it's just the possibility of having alternative pathways that makes it so challenging for the treatment of some of these diseases. Now, ideally, we would like to find a way where we can find such a powerful way to trump a disease, an Achilles heel, like this lamppost. If this wire, telephone, electricity, etc., if you're able to bring that down, you're creating chaos in the neighborhood. So we would like to have opportunities like this as we go ahead and treat cancer. And the reality is there's very few cancers where we have that. In the disease I deal with, myeloma, we've known for about 20 years now that they have chromosomal translocations, one of which is this gene, involves gene FGFR3, which is associated with this 414 translocation. Well, and we know that in vitro and in vivo with some xenograft models, you can actually make the cells disappear. But guess what? We tried this drug in patients and it didn't work. This is a bona fide target for us to think about individualized medicine. Now, unfortunately, in this example, you have a gene that's juxtaposed to a very powerful genetic enhancer, the IGH enhancers. So trying to shut that down was like trying to shut down the oil leak that we had in the Gulf of Mexico. You need a very powerful way to do that. If you don't do it completely, the cells don't die. Not suggesting it's not a good target, it's just that we need to get better at approaching how we're going to plug those translocations. I'll use another example from the disease I deal with, again, myeloma. As you know, the sort of fairy tale story of individualized medicine is the development of imatinib for the treatment of chronic myelogenous leukemia. And that has led to a number of second and third generation drugs. Now we have things such as ponatinib that can overcome some of the mutations that are associated with this. And it turns out most patients are functionally cured from their disease when they take this type of medications. But if the disease is a little bit more advanced, then it's not a very good treatment. So this is a very important study came out from China, where they looked at the next phase from what we call chronic CML. It's called accelerated phase, which is just the step before acute leukemia, which is a natural progression, CML, accelerated phase, acute leukemia. Well, they tried imatinib in these patients, and lo and behold, classic bone marrow transplant was much better for them. So it was a little bit too late to think that a small molecule in and of itself would actually do the job. In fact, we use this drug in the setting of hematologic malignancies, but we have to do so in combination with chemotherapy. And this may be something similar to what we might see in many of the cancers we're dealing with. Now, I'm going to show you a detailed example of how we're doing this for myeloma patients. So the timeline in the bottom is, as the patient was treated with multiple regimens. What you see in the red and the blue are the markers that we use to measure sort of response to treatment or not. So the patient gets a course of treatment first with lenalidomide and low doses of dexamethasone, does very well, and then gets a series of treatments, including proteasome inhibitors with carfilzomib, bortezomib, goes back to alkylators, multi-combination chemotherapy, and ultimately the patient dies because of the disease. Now, we have the opportunity at multiple time points to collect samples from the bone marrow and the blood of this person. And this is what we found. If you take a genome-wide view, so this is diagnosis in blue, relapse in red, you see that the genomes are actually quite similar. But you start seeing some differences. So this is chromosome 1 at the far left, 2, 3, and so forth until we get to X and Y. If it goes down again, it's a deletion. If it goes up, it's an amplification. And as you see, for instance, in chromosome 1, there's an amplification that was not present before, and the deletion that was present at diagnosis is no longer present. Well, you know that DNA cannot reform itself. We cannot reform whole chromosomes, so that means there was a component of separate clones actually present at the time of diagnosis. If you drill down into the markers, for instance, you find some markers that are persistent at diagnosis and relapse. But then again, you go back to another one that I showed you, that a patient had a deletion at the time of diagnosis, which is actually gone at the time of relapse, a whole segment of that chromosome. But now you have a separate one down the line. So there's some changes that tell you that there's a possibility of multiple clones. And again, if you drill down even further, you see on the example on the left where there's that shift to the left on green, that at diagnosis, you already see there's a faint sort of color green that is going to the left, but then that becomes very evident at the time of relapse. One of the genes that is in that region is BRCA, which is associated potentially with refractory state to bortezomib, one of the drugs we use. And this is a summary slide of all the testing that was done in this patient. It was extensive genomic testing. What we find, and they are all represented by different colors, is that the patient had multiple clones. At the time of diagnosis, you see that clone 1 and 1.2 predominate, the orange and lighter orange. The patient goes on treatment, and the clonal composition looks similar, but ultimately, clone 2.1 emerges as it is refractory to such treatment. The patient is switched to a proteasome inhibitor, and then we go back to clone 1.2. And then with further evolution of the disease, you see that ultimately, the blue clone, 2.2, takes the life of the person. And in fact, that was present right from the beginning. So it's sort of a selection through administration of some of the therapeutic drugs, but in fact tells you about the multiple pathways that cancer can take and overcome some of these targeted approaches that we so much want to develop. So for this, I use the story of the lion and the gazelle. And you've probably heard this African story that every morning, the gazelle start thinking, well, you just have to run, but I just cannot be the slower gazelle. And the lion thinks, well, I have to run fast, but not as fast as the fastest gazelle. You just need to catch the slowest gazelle. And that may be what we're doing sometimes with chemotherapy. If we are to win, ultimately, in the war against some of these more complex cancers, we've got to get rid of all the gazelles right from the get-go. We cannot let some gazelles go on because over centuries, the gazelles always win. So I'm going to show you just some practical examples of what we have done in the clinic, real-time application for next-generation sequencing for the clinical practice. And I've talked already about some of the challenges that we have. We began a pilot study for this in the third quarter of 2010 and tried to address some of these challenges, and actually began, as you've heard through this conference, with the application of a tumor board to provide some context to result interpretation. We actually were able to open 2 protocols approved by the Mayo Clinic IRB, one which was institutionally funded so that patients could have access to this sequencing through institutional support. And then, through much discussion with the IRB, we got a unanimous approval for a self-pay protocol, where for selected patients, patients could pay for the genomic cost of doing the sequencing. This involved, of course, all the safeguards, including consultation with genetic counselor, and a lot of the redundancy that was needed so that this would be done in a proper fashion. And this was the criteria. Patients with advanced malignancy wanted a life expectancy of about 3 months. Now, a lot of this, at the time, we did in collaboration with our partners at TGen and have done a lot of the work, of course, with the Center of Individualized Medicine, where I won't take you through all the steps, but as you know, we do this, the biopsy, we do the sequencing, we do the validation through the CLIA, and then we come to a consensus board where we recommend treatment for our patients. So I'll show you 3 quick examples. Cholangiocarcinoma, a patient with fairly advanced cholangiocarcinoma. We did the next generation sequencing. What I show you here are the 3 lanes of sequencing. At the top is germline. In the middle, you can see that with DNA sequencing, we found a mutation, but only in a few of those lanes. You can barely see that in green in the mid panel. In the lower panel, you see the value of transcriptome. This RNA was expressed at high level, and we could detect the mutation readily. This mutation of this gene that you see, ERRFI1, resulted in constitutive activation of MAP and AKT. You can see that by immunohistochemistry. Again, I won't go through the details, but I'll show you the clinical results. So this is a patient that at baseline had a mass there juxtaposed to the spinal vertebral body on the right. And then you can see how that has essentially disappeared in the PET scan on the lower part. Now, some of those results have not been definitive. Some of those patients have experienced the recurrence of the disease and so forth, but at least shows proof of principle that this works. And a very interesting part is erlotinib, which is the treatment that we use for this patient, had been tried globally against cholangiocarcinoma, and it didn't work, but it did work for this specific patient. Dr. Carpton presented these results at the meeting where he actually showed that a fraction of these patients also have a translocation that involves FGFR3, just as I showed in myeloma. And these patients, in fact, expressed by immunohistochemistry augmented levels of FGFR3. Most importantly, these patients were treated with a pan-kinase inhibitor, ponatinib, actually show tumor regression. You see the lesions right there in the mid-abdomen become cystic and necrotic with administration of ponatinib. And this was, again, just a reflection of the targeted therapy guided by the whole genome sequencing. 2 more quick examples. Cervical cancer, a patient that was referred to us by our local partners. This patient had a relatively stable genome, but ultimately were able to show HPV-18 integration into chromosome 6. So the prediction would be that more and more cancers where there might be a viral association, you would be able to pick that up through next-generation sequencing. And the last example, of course, I couldn't do so without showing you a myeloma slide. Patient with extramedullary myeloma. Now what you see in the center are all this chromosomal rearrangements and translocation. So it just gives you an idea of the complexity of a case like this. I won't show you all of the specific examples, but I'll tell you the genes at the very top, cereblon, associated with resistance to thalidomide and lenalidomide the patient had. The next 2 genes are associated with resistance to steroids the patient had. And the bottom one's associated with resistance to proteasome inhibitors. The patient was resistant. So it was all consistent with what we were seeing in the clinic. To date, 40 patients have been enrolled into these protocols. The results are complete on 33 patients, which includes 6 from this pilot phase. And in 16 patients, not always you get a target, we have identified 34 potential targets. 80% of them have been validated, and the only reason it's 80% is because the translocation ones has not been an outlet yet to validate them, but all the mutations have been validated. As of this work, 10 patients have been treated because of the results with next-generation sequencing. 4 patients have no actionable targets, and as would be normal in this type of scenario, 4 patients expired prior to the availability of the results. The turnaround time now is in the order of about 2 months. Oops. Could I go back one? This is just a list of some of the drugs and the targets and the various cancers that have been treated, some of the responses that we have seen. But I think this very last slide shows the commitment that Mayo Clinic has to try to bring this to patients. There's a single patient IND for a drug that was available for what we thought was an actionable target. So, again, many challenges. We're trying to develop a protocol called RAPID, where in collaboration with various pharmaceutical companies, we could have a predetermined pathway for having access for some of these drugs that are not currently FDA approved for some of these cancer indications. And again, we'll continue to work closely with the rest of the Center for Individualized Medicine and the components that it has, including bioethics, genetic counselors, et cetera, so we do this in the proper way. To conclude, I'll tell you, I think we're past the stage where genomic medicine was seen sort of as a scare, and in reality, there's much more promise than we actually were ever made believe. It's just an incredible opportunity that we have. And for anyone who's a resident or a medical student, I'm jealous to see what kind of medicine you're going to be practicing 30 years from now. I'm going to close with these two slides. The first one is, on the left, you see the Nautilus, of course, submarine, historic. Back then, I'm sure when they were building it, they were thinking about the thickness of the glass for those windows. It was just necessary that you had windows. That's how you navigate in the world. Well, I think if you propose to put windows in a nuclear submarine, you would be probably booed out of the room, because we navigate through different tools. And I think that's what's happening. And we might see a revolution where pathology may go from more of the visual inspection, probably there'll be a component for that still, but more towards the genomic profiling and understanding of the tumors and the diseases we're dealing with. My last slide is this one, which I would encourage you all to read this book. It's called Bad Medicine by David Wooten. I won't spoil you the book by telling you what the premise is, but he shows with data that in the history of medicine, more harm has been done by delayed introduction than by haste. And I think that's one of the challenges that's upon us with genomic medicine and application this to the bedside. Clearly, we have to be deliberate, careful, but also we don't want to be holding things that potentially are of benefit to patients, as I was just shown in the previous slides. I want to acknowledge, of course, everyone at Mayo Clinic, in particular Dr. Bryce, who helped me put some of the slides together, hopefully to prove the point that, in fact, genomic medicines can be of clinical benefit. Thank you.
Rafael, thank you very much. Again, just to remind you, as questions arise, if you would begin to direct them up, and we will be taking them at the end of the session. I'd like to introduce Dr. Charles Erlichman. Dr. Erlichman is the Peter and Francis Georgeson Professor of Gastroenterology Cancer Research. He is the former chair of oncology here at Mayo Clinic, and he's had extensive involvement as it relates to developmental therapeutics, both in terms of being leader of the Mayo Clinic Phase 1 UO1 cancer grant for many years, the Mayo Phase 2 Consortium. These are NCI-sponsored grants, and also as it relates to the North Central Cancer Treatment Group and now the Alliance for Clinical Trials. So Dr. Erlichman, thank you.
Thank you, Matt, and good afternoon, everyone. Thank you very much for the opportunity to participate in this exciting symposium. I'm going to be talking today from the perspective of an academic who has worked in drug development for many years and looking at the issues of challenges and strategies that we can approach in bringing new agents into therapy during the genomic era. And part of what you've heard from Dr. Fonseca will be related to some of the things that I will make reference to. So every morning, I can wake up and read the Wall Street Journal. Some days I get an opportunity to determine that I'm going to have a revolution in my cancer treatment, so that maybe I'm going to be out of a job. And of course, there are the anecdotes that go along with these sound bites saying, this is the greatest information. And yet, maybe all I'll need to do is call up somebody's genome and prescribe medication and it'll be a solution. Well, we all know it isn't really that easy yet. So what I'm going to do over the next few minutes is talk about some of the challenges. And the challenges are listed here in two colors. And the reason for the two colors is that the orange ones are the ones that I'm going to focus upon. The others not because they're unimportant, but because there's only so much time and only so much knowledge that I have, will not be addressed in any direct manner. I will then talk a little bit about strategies, talking about general trial designs, give you a couple of examples of what the NCI is planning, what one of the cooperative groups is planning, and then also talking about strategies that are personalized, but not necessarily as genomically oriented as one often thinks about. Start off with this Rare Disease and Orphan Drug Act, and you're wondering, why in the heck is he talking about that? Well, the Rare Disease Act basically just defines what a rare disease is, and it was legislated in 2002. The Orphan Drug Act obviously dates back even further. That Orphan Drug Act was built done in order to facilitate and incentivize drug development in rare diseases where a financial return for the company doing this was going to be unlikely. And you can see all the things that potentially could come up in if you got an orphan drug status for your agent from a company perspective. The important point to realize or acknowledge is that these two acts were passed before anybody thought about genomics. So a rare disease is anything that's 200,000 that affects 200,000 people or fewer. And similarly, it's for the drugs for diseases that affect 200,000 people or fewer. Okay, so that means breast cancer is a common disease and not rare. Lung cancer is a common disease and not rare. But if you start saying, what is a genomic subset of lung cancer? Is it a rare disease? Should it be looked at in the context of drug development for orphan status? These are issues that actually need to be considered, and I'll show you what the consequences of this is. Let's use two examples. One is a practical one, one's a hypothetical one. Lung cancer has the incidence of over 200,000 a year in 2013 is what's expected. ALK-positive lung cancer at most probably consists of about 7% of that population, so that's 16,000 people. The registration trial that got crizotinib approved ran between February 2010 through February 2012, and a total of 4,967 people were screened over a period of 2 years to randomize patients to 347 patients to this trial to demonstrate the clinical activity so that FDA would give the approval. Okay? That's not too bad numbers-wise, although you've got to exclude over 4,500 people. Sorry, sir, you cannot participate. Okay? That's one genetic abnormality. It happens to be a translocation. Let's pick a hypothetical tumor that has two genetic abnormalities, and for the purpose of this discussion, it'll be two driver mutations. So if you've got driver mutation 1 and it occurs in 2% of the population of tumors, the next one, this driver 2, or mutation 2, occurs in 8% of the population. You then factor in an eligibility factor, because there are many eligibility criteria in getting onto a clinical trial. And let's say, conservatively, 50% of the people would be eligible if you didn't count the mutation part. And then finally, there's the fraction consenting. Depending on the nature of the trial, a lot of patients will not want to go on a trial that is randomized, for example. And I'm giving that fraction consenting an 80%, meaning 20% would refuse, which is actually quite low in usual clinical trials. There's another factor which I haven't put in there, and that's the doctor or the caregiver. Because if they don't promote the trial, then patients just don't necessarily participate. But if you just do the simple math, and this is, I'm not a statistician, and there's probably statistical flaws in this analysis, but if you just do a rough analysis, what you find out is you now need to screen 1,400 patients to register one subject. Wow. That means a clinical trial needs to be very big in terms of access to the population. It now is becoming population science, not clinical trial science only. And then it begs the question, should treatments for the subsets of common tumors be given orphan drug status? Another issue is the issue of single or multiple genomic alterations. We all know, and you heard from Dr. Fonseca, there are mutations, there are amplifications, there are translocations, there are deletions, there is silencing that is going on in genes. This is all happening. And we do the analysis. The technology is there to do that analysis, and that's great. And then what do? We say, which ones are the actionable targets? That's the targets for which there are drugs, or we think there are drugs out there, or agents. But do we know if those actionable targets are truly drivers of the cancer, meaning the cancer is dependent on this abnormality, or it's purely a passenger? Not necessarily. We're hoping that it is, but we don't know. What is developing are the bioinformatic methodologies to look at pathways and networks so that we can hopefully understand. And when you recall Dr. Fonseca's poll and bringing that down, hitting the nodes in a network, multiple nodes will bring the whole network down. We're still early in that development. But there's two other things that are lurking in the shadows, one of which Dr. Fonseca alluded to, and that's the tumor heterogeneity, the multiple clones. And the other thing is the dynamic nature of the cancer. The cancer doesn't just sit there just because it's got a bunch of clones. It's changing constantly. So now you've got a mixed population, and that mixed population ain't staying the same. Well, the solution to that problem, obviously, is you get tissue, right? And you make the analysis, and you've got a solution. Well, originally people started thinking, well, you get tissue from the original diagnostic biopsy or surgery. And there are problems with that. First of all, if the patient had breast cancer 20 years ago, can you find that tumor specimen wherever it is? Secondly, the institution that has it may not want to release it for various reasons. Thirdly, when they collected the sample 20 years ago, how did they collect it? Did they collect it in a standardized manner so they could be processed and you know the provenance of it? Or was it collected and then thrown on a shelf where the temperatures went up to 90 degrees one day, minus 20 the next day, and now you're going to analyze this tissue? And then, as I've already said, the primary tissue genomic characteristics are not necessarily the same as the metastatic lesion 20 years later. So the biopsy specimens, even if you get them, may contain different clones of tumors with different genomic characteristics. And excuse me. So to overcome that, the solution is we'll do biopsies now. Patient walks in the door, we'll do the biopsy now. That overcomes that theoretical concern. The samples then can be collected and processed and analyzed in a standardized manner that's predefined. But then will the patient agree to research biopsies? Some will, some won't. And then who will bear the cost? As you heard, for drug company studies or studies that are for drug development, the drug developer will have to foot the bill. Outside of a trial, it's a question of will the insurer pay for this, or will the patient have to pay for this out of pocket currently? To show you this slide, and again, I thank Dr. Fonseca for showing the schema, but the point I'm trying to make here in this context is that you've got multiple clones, so if you stick the needle in the wrong place, you may pick up the wrong clone, in which case you may not be targeting the right thing. Similarly, each time you treat the patient, you change the clonal distribution, or you will run the risk of changing that. And therefore, you may not be dealing with the same thing. Does that mean every time the patient recurs or progresses, you need to biopsy again and then hope to heck that you're going to get the right clone that's driving the tumor? Those are some of the challenges that we face. So what are the strategies to overcome these challenges? Because when you start thinking about this, it becomes, oh, I need a Tylenol for my headache. So there are two general approaches that are being taken in drug development currently. One I've referred to as the umbrella trial, and this is where patients who are eligible for the trial agree to undergo tumor biomarker analysis. Notice I say tumor biomarker analysis, not just a genomic analysis. And then through a bioinformatic algorithm, you determine which biomarker putatively drives the tumor. And the patient is then assigned to biomarker A, B, C, D, or because they don't fit any of those biomarkers, they're classified as no biomarker. And then you then assign them to a therapy that's matched to that biomarker, hoping that you're picking the right biomarker and the right therapy. And the patients who have no biomarker are assigned to standard of care therapy, or they could be randomly matched to any of these treatments. And they function as the control because they're getting the unselected treatment, okay? So this just summarizes the design. You characterize the tumor genome. There are multiple treatment arms based on target and drug pairing. You can select one tumor type. You can do this in breast cancer. You could do this in lung cancer. You can do this in colon cancer. You could even do it in myeloma. You match the tumor to the drug based on some sort of analysis. And then you treat the patient, and if there's no benefit, you have the potential to say, okay, it didn't work. Maybe I picked the wrong biomarker, and they have another biomarker, so you now can allocate them to another one. That's the sort of approach that you can take. The question being asked in this type of trial is, does selecting patients for a treatment based on a biomarker increase the likelihood of benefit? You can build in what's called an adaptive randomization so that as the data comes up, you say, okay, well, so many it worked in, so many it didn't work in, we'll drop that and we'll move in a different direction. All patients can participate. It doesn't matter whether you have a biomarker or don't. You can still participate in the trial. There are many different agents that are available, and you can add and subtract agents. You can say you want to see a certain level of activity up to a certain point. If it doesn't hit that after you've treated 20 patients, 30 patients, whatever the number is, you can say, we'll close that arm. We're going to bring a new drug in, okay? So, and finally, because this is a very practical issue, the way we practice medicine, I'm a medical oncologist and I deal with GI cancer. Dr. Fonseca deals with myeloma. You can focus on a specific disease and get the specialty area and the patient community engaged in that research, and that promotes and facilitates the study. There are some limitations, and I've already mentioned these. It assumes that the genomic change selected is the driver. It assumes that you know the target of the agent. In other words, if drug X works on target Y because that's what FDA approved it for, or that's what it was developed for, are we sure it doesn't work on 6 other targets? It probably does. But do we know which ones? We're making an assumption. You may need multiple biopsies because of the genomic heterogeneity and instability that I've mentioned before. And then you have a challenge if you want to do these studies, because you may need to get different drug companies with different interests participating. This can be a real challenge because no single drug company has a pipeline so rich in clinical trials that it can hit just about every target that's been described as potentially a driver in the process. Well, as soon as you start putting drug companies together, then you have to deal with issues of IP, which I referred to in my first slide, but I'm not going to get into further at this point. So here's an example of how NCI is approaching it. This is a planned study that they are establishing. This study is not up and running. It is currently in the discussions with FDA. But basically, it is an umbrella study or an umbrella protocol which will have multiple single-arm phase 2 trials, each with molecular subgroup matching to a targeted agent. The agents can be commercially available or experimental and would have at least phase 1 safety data so you know what the right dose to try is. And NCI will act as the drug pharmacy. In other words, it will engage the companies and work as an honest broker to facilitate the availability of these agents instead of having companies trying to play off of each other. And then the arms will be, there will be an IND and IDE for the protocol for the treatment and the diagnostic components set up for each agent using a template approach with FDA. And there's a standard bioinformatic analysis that will be done across all arms for all patients. As I mentioned before, the arms can be deleted and added to. And initially, the plan is to use single agents and then consider combination arms. By using a central IRB nationally, then hopefully this will facilitate the trial activation and moving forward on a regulatory basis. I'm not going to walk you through this algorithm, but this is a schema for a patient going on the trial. As you can see, it is complex, it is very involved, and requires multiple expertises to come up with a treatment option for a patient. What does this mean for screening? NCI is planning a screen of 3,000 to 4,000 patients to enroll 1,000. They are going to hopefully select, are planning to select labs running comparable screens for these different mutations, mutations, I should say, under a particular IND and credential the sites. They're going to limit their enrollment right now to 1,000. I think that's probably has something to do with the fiscal viability of our U.S. government. And they're going to try and cover common and rare tumors. And this is an outline of the approach they're going to take. Now, what I want to show you here is another example of an approach that's being taken. It's another example, actually, of an umbrella trial. This is a trial done by, being developed by the Southwest Oncology Group Lung Cancer Committee. It is for squamous cell lung cancer, second-line therapy only. And you can see how complex it is just because of all the number of arrows and arms, et cetera. But the point I'm trying to make is, if you look here, again, biomarker assigned, and then in this case, they're going to randomize to either standard chemotherapy or a targeted agent based on the biomarker, each one, okay? And then they're going to get to a certain point and say, well, is this active or not? If it's not, they'll just drop this out and replace it with something else. And they will have up here the non-matched drug or standard chemotherapy for the control arm, and it allows all patients to participate. At the last discussion that I had with the people organizing this, they had 12 companies sign on to make their drugs available, and they're hoping for more. The other approach is what I'm referring to as a basket trial. In a basket trial, things are sort of reversed. In this case, you don't care what the tumor is. You're really focusing on the the pathway or the target. So you have here a bunch of different tumors. You take those tumors and you do some sort of biomarker analysis for a particular targeted therapy that you've defined for whatever reason, and then treat. So that you're asking a different type of question. The question is, does hitting the target result in an anti-tumor effect? It assumes that a target or pathway is shared across many tumor types, which is becoming more and more apparent, but not consistent. It assumes that the target will be a driver across many different tumor types, and we know that ain't necessarily true because, for example, BRAF mutations, yes, in melanoma, BRAF mutations in colon cancer, well, I don't know. So you assume that you understand how the therapy works. Again, that's always an assumption that we make when we treat patients. Patients who may respond, even though the biomarker is absent, will be overlooked because if they don't have the biomarker, they can't even participate. Now you've got to go back and ask doctors like Fonseca, Erlichman, Getz, who treats breast cancer, will you all participate in this trial? And each of us are in different communities, and it becomes more of a challenge. The advantages are there are many different tumor types that can be enrolled. There's the potential, again, to add or remove tumors, remove tumor types. You do not need multiple biopsies. You just do one biopsy at the beginning of the trial. The limitations are that you won't be able to allocate all patients. You need the cooperation of many specialists. You assume that the genome changes selected is the driver again, and you assume the target of the agent is known. Areas where this approach actually is being explored and probably being very good is something that we don't think of commonly when you think about genomic medicine, and that's antibody conjugates, antibody drug conjugates. In this case, you're looking for a particular epitope expressed on the surface of the tumor. Okay? That epitope is targeted by an antibody, regardless of what tumor type it is, as long as you have that epitope, and the anti-tumor agent destroys the cell. There are 15 companies with at least 30 ADCs in development currently. And the other area is immunotherapy. Affecting the immune system may have an impact on a number of tumor types, and the classic example today is anti-PD-1 and anti-PD-1L strategy, where they're looking at multiple different tumors targeted by one strategy. That is only the beginning. So I'm going to end up by reminding you a little bit of history also. Anti-estrogen therapy, meaning tamoxifen, was first reported in 1973 as being active in breast cancer. At the same time, the biomarker assay was reported in 1973. So both are 73. We learned from this, or we thought we did, and then along came Herceptin and HER2. And it was almost like we didn't learn anything from our 1973 example. So we need to keep that in mind, that A, we've been practicing individualized medicine in cancer at least as far back as 73, and B, that we don't need to reinvent the wheel all the time, but take things from before and apply them and build on them. Thank you.
Just like to thank you, Dr. Erlichman, for bringing it back to the point that it's the breast oncologists that know how to practice medicine. So, all right. Well, with that, we'd like to welcome up to the podium Dr. Taylor. James Taylor is an assistant professor at the from the Department of Biology and Department of Mathematics and Computer Science at Emory University. And he's one of the original developers of the Galaxy platform. I think you're going to be talking to us a little bit about that today. He's also actually been someone who's really promoted this idea of having systems that can be extended across different institutions so that we can actually reproduce what other people have actually have performed, which, by the way, is a very big issue. And so with that in mind, we're very pleased to have you come and give a talk. Thank you.
Thank you. And thank you very much to the organizers for inviting me to speak here, given that I know nothing about cancer care whatsoever. But I will do my best to try and give you something entertaining and informative anyway. What I do know about is sequencing, and so right off the bat, I need to tell you how much I love sequencing. Sequencing has essentially been either the driver or the enabler of my entire research career, particularly high-throughput sequencing, and so I'm now going to say lots of bad things about it, and don't let that dissuade you from believing that sequencing is or will someday be the solution to all of your problems. All right, so why do we love sequencing so much? High-throughput sequencing is clearly transformative. I don't know how hard I need to bang on this argument anymore. Mostly for what I'm talking about here, we'll be it's either resequencing or direct transcriptome sequencing. But actually, most of my research interests are down in the bottom here, all the interesting stuff you can do in terms of reading out transcription factors, reading out the 3D architecture of the nucleus. So sequencing has really become this technology that's enabling across all areas of biological inquiry. So the other great thing about high-throughput sequencing is it's democratizing. This is the map from omicsmap.com yesterday. And this is basically telling you numbers of high-throughput sequencing instruments all over the world. So this is a widely distributed technology. It's also essentially free. And I hate to show this slide that everybody shows, but I've heard throughout this meeting some talks about some discussion of the cost of sequencing. Well, this is log, right? This is exponential. If sequencing isn't free today, it will be free before you know it. The costs of upstream prep and the cost of downstream analysis are going to completely dwarf any cost associated with sequencing itself. So this is fantastic, right? Just wonderful. So not only is sequencing inexpensive, widely available, broadly applicable in almost any area of inquiry. So we're seeing individual investigators as well as, obviously in the clinic taking advantage of these technologies. So how's that going? So just to back this up, we have published on this, and so the data behind what I'm going to say is available. So one of the first things we did, and this is mostly in the research context right now, we're thinking about translation. Obviously, we start in research. And so these are all resequencing studies. These were done in this was prepared about 3 years ago. And so what we basically did is ask, okay, were we able to get the data? Were we able to actually get the analysis methods? And were we able to get all of the information to take those analysis methods and apply them? Short answer, no. 0% of these studies could we actually reproduce what the authors had done. We moved forward to 2011. Now, suddenly, resequencing has taken the world by storm. BWA is one particular tool that's fairly widely used for aligning short sequencing reads back to reference genomes. There were about 380 papers published that used BWA in 2011. We took a random sample of 50 and found that a number of them did not provide primary data. This is particularly shocking. Then others provided various amounts of information. But at the end, only 7 of these papers, better than 0%, but still, only 7 of these papers provided the details necessary to reproduce these analyses. Do we care? That's a question I'm often asked. So what? Well, these details do matter. So this is one example. This is actually this belongs in the other OMS section because this is a mitochondrial heteroplasmy example. But what we're seeing is the level of a heteroplasmic variant, and across the x-axis we have different versions of the tool BWA, and we have different parameter settings. And as you can see, the results that you get vary wildly depending on these simple details that nobody is actually recording. So that's one issue. Now, this is a really nice paper that came out in Genome Medicine back in March, and the punchline is in the title: low concordance of multiple variant calling pipelines. So this was a really, really nice study. They did exome sequencing, so that using the SureSelect2, and 120x mean coverage. So this is deep sequencing. Way beyond, most people rule of thumb sequencing are doing 50, maybe 60 if you're being conservative. So double that, and then took 5 common variant calling pipelines that are being used all the time in various places and applied them to this really rich data set. And they produced the most beautiful Venn diagram of all time, which is why I love this paper so much. And you see in the middle, 57.5% concordance between these variant calling pipelines. And this is the state of the art of variant calling in next generation sequencing. Every single one of these pipelines called some number of unique variants. Every single combination of 2, 3, or 4 pipelines called some unique combination of variants. And that's just for single nucleotide variants, right? And this should be the easiest problem in all of sequencing bioinformatics. We have a reference genome that's the best quality reference genome that's ever been produced. We have incredibly deep, high-quality sequence data. This is the best we can do for single nucleotide variants. Only 3 of these pipelines will even do indels, and for the 3 that do indels, they have a whopping 26.8% concordance in their indel calls. Okay. So we are far from establishing reliable best practices, even for the simplest problems. My claim is the democratization of sequencing enabled by high-throughput sequencing instruments has not been matched by democratization of informatics techniques, and yet these techniques are absolutely required to make sense of this data. And the question we've been asking is, how can we ensure these methods are accessible while also ensuring results are reproducible? So that's where Galaxy comes in. I'll just talk briefly. This is the system we've built. It's a web-based system for performing genomic analysis. The idea is that you can easily use complex tools, and yet every detail of the analysis is automatically and transparently tracked. So provenance is no longer an activity. It's just something that happens automatically. The other piece is that all data is immutable, and nothing is ever deleted. And so what this means is you have complete provenance, complete transparency, and complete reproducibility for all analysis. From various levels of granularity, we have where you have your tool developers basically packaging up the software that they've developed in an abstract way. We then can build the analysis environment on top of that. We build workflow systems for more complex analysis, and then even higher levels of abstraction, like entire publications that have all of the data and all the details integrated. We do interactive web-based visualization. So each step of the way, we're pushing higher levels of abstraction from the very, very raw data to something that's aggregated and something that's navigable at a high level. And yet all of it's connected, and nothing is ever lost. Since it's cancer, I'll try and give you an example of how we're applying this platform. And so this is all work by Jeremy Gex, who was a postdoc in my lab and just took an assistant professor position at George Washington, and with Basil L. Reyes and Mike Rossi, who are in medical oncology, radiation oncology at Emory. And so what we did here, this is just a pilot, but we took 6 patients and did whole transcriptome sequencing of primary tumor mixed. We also did this analysis for this MIA pancreatic cancer cell line, did whole transcriptome sequencing there. We also did deep exome sequencing that's not mentioned here, but which we'll use as a validation measure, and so we get a substantial amount of sequencing data. We first had to integrate some new tools for discovery. So these are not tools that we wrote, right? These are tools that other people had made. Veriscan, in fact, coming from the Washington University group. Top Hat Fusion coming from the Maryland group. All sorts of tools for variant annotation, working with these variant files. And then we build workflows to actually analyze this data. And so this is a single workflow where we actually get out gene fusion information, transcript, expression level information, and we're calling variants from RNA-seq data. This scared me at first, but I believe we can call variants from RNA-seq data. One of the ways we validated this was actually using the extremely deep exome sequencing as a control. And if we require greater than a 7% allele frequency on our RNA-seq calls, we get a balanced false positive and false negative rate of around 5%. And that's probably pretty good. Scary. We've also built pipelines for comparing with public data sets. And so this is an example of comparing against the Cancer Cell Line Encyclopedia. How much time do I have? OK. So this isn't fantastic, right? So we have our 6 patients and we have our cell line. Wonderfully, our cell line, so there's 4 mutations in OncoMap in the cell line, and we find 4 mutations. The patient's mostly 0. Interestingly, so patient 1, 2, and 3 have more mutations in common with the cell lines than patients 4, 5, and 6. We went back and kind of looked at that, and one of the concerns was, well, what's the tumor percentage? And so we went back and asked, and it turned out someone had actually looked at that in the meantime and had said, oh, I think that one's actually 0%. But that's totally wrong. We can see these mutations in this patient 4. And so in this case, the genomic data is probably more accurate, but nonetheless, you see that the first 3 patients are much much more tumor than the second 3 patients here. We can then build workflows to do clustering, and you'll see, oh, wow, look at that. Patients look like each other and don't look like cell lines. This is Pearson or Spearman correlation. Cell line correlation is actually negative. So this is harder. Yeah. So these groups cluster together, and there's some reasons why that might be, tumor percentage being one of them. Big workflow to do integrated analysis of variant gene expression and annotation. And so now we can use differential expression to pull out our top candidates. We can annotate them using ANNOVAR in terms of what the impact of the mutations in those top candidates may be. And we're pulling out some interesting stuff from this. There's actionable stuff here. I don't have a story yet, basically, because this is kind of where things end. This is what we can automate, and now it's basically Jeremy and Basil looking at genes and variants and being like, oh, maybe we can do something with that one. But I will mention we have the power to use visualization to drive our ability to do better analysis. And so this is interactive visualization of the variants for these 6 patients. We can actually reanalyze data on the fly. So if we want to rerun our variant annotation with different parameters, we can do that in the context of the visualization. We can go farther than that. We can actually do parameter sweeps to try and set parameters, because the reality is we have no idea how to set these parameters, right? That's the problem. We don't know. We don't have good gold standards, and we don't really know what we're doing. And so using visualization is one really powerful way that we can try and do better. So finally, we're interested in matching patients to cell lines. The nice thing, so these are, the highest matching cell line in the Cancer Cell Line Encyclopedia for all these patients is actually a pancreatic cancer cell line, which surprised me. The number of mutations shared is pretty small. So my conclusions. The important conclusions are that we can build precise pipelines enabling complete transparency and reproducibility analysis. This is an open platform. It's completely open source. It's openly developed. And so the idea is that we can actually share and publish these analysis methods so that other people can take them, translate them into their own environment, and precisely reproduce analysis. And what this allows us to do is actually kind of crowdsource the development of best practices as we push forward and try and figure out how to do bioinformatics right, because we're pretty far from knowing the right way to analyze this data. From a practical perspective, I've shown you that we have a variety of workflows for doing some interesting things with cancer genome data, and those we can make available and are making available, and other people can use because of the nature of this platform. Problems. Numbers of samples, numbers of known variants in public databases is far too small. We need 1 million cancer genomes. Let's just do that. But also better annotation. So there's annotation sources out there that we can pull in for genes variants, the druggable gene databases. And what we're thinking is actually building high-end interactive data mining tools to let people dig in there and try and find these actionable targets. And that's it for me. Just again, acknowledging Jeremy, who did all the work I talked about in the second part, my collaborators on Galaxy. And thank you. Thank you.
Well, we're going to begin a time of discussion. And as you can see, the speakers were brought together for this session for a reason. And the reason is that you can see is the complexity of doing genomic and individualized medicine, and the complexity of really the issues with doing it well, but also the promise. So I think that as you think about genomic medicine, I sort of think about it as a sort of a U-shaped curve, and that is that at one end there is the issue of incredible promise. Everybody thinks this is the way, and then ultimately when we get into the clinic, we begin doing it, you can actually become overwhelmed and think, this is just never going to work. But I think there's somewhere in between, and that in between is that there are clear evidence and clear examples that it has worked. But yet there are challenges, and how do we go forward? So with that in mind, we actually have a series of questions. First, to start out with, it's audience response. So if you have your ability to actually go to your smartphone, and you can see up here, our first question is, should all patients diagnosed with cancer have genomic testing? And A A, at what stage? B, should they have sequencing performed at several time points? And C, are some cancer types better suited for genomic testing than others? So number one, should all patients diagnosed with cancer have genomic testing? And our possibilities are all of the above, a boon because failed phase 3 drugs may be resurrected for certain groups of patients, a bane as we'll never find enough patients to satisfy the current statistical paradigm for validation studies. All right, so this is our answer, all of the above. We're going to actually keep going with the questions, and then we'll come back to these. So question number 2. We're going to move on. Can we go up to question number 2? We're still waiting? Okay. I have a disconnect between my questions up here and those up there. So let's go with what's up there. All right. Okay. Let's go with this question, and this is what we're going to focus on right now. Number 3, which of the following will become the first standard of care for cancer patients? Okay, so tumor exome sequencing and interpretation, tumor genome sequencing and interpretation, none of the above, tumor transcriptome sequencing and interpretation, or one or more of the above. Continue to provide your input here. Okay, is that our last question, or do we have one more? Okay. Let's go back to number 2 because we kind of skipped over this because I had it on different on my screen. I think this is a really important question. It's one that we're faced with all the time. It's certainly one that I'm faced with my patients. Is the N of 1 realistic for cancer genomics? Yes, that's what individualized medicine is all about. No. Cancer patients will still be stratified just by different criteria. You're uncertain, or both A and B. All right, continue to provide your responses. Okay. All right. So these actually questions are very important as we think about bringing this technology forward into our patients. And as I mentioned before, you can see some of the challenges. And as we're beginning to think about these challenges, we're going to actually begin by bringing forward some of the questions that each of you have. And I'm going to start by just addressing one practical question, and that is just the cost of doing a biopsy. So when I'm seeing a patient who has metastatic breast cancer in the clinic, and that particular patient has, let's say, progressed on a certain treatment, one of the questions that comes up, and that is, should we do another biopsy of that patient? So in the era of genomic medicine, there are certain cancers where you actually have literally very few, if any, options. So doing a biopsy is not necessarily going to be covered by an insurance, especially if you wouldn't necessarily be using that information otherwise. But there are situations and scenarios where actually now we're beginning to see the impact of doing biopsies on multiple times. And I'll give you an example, and that's in breast cancer, where actually there's data that demonstrate that knowing that, first of all, that the estrogen receptor, which Dr. Erlichman referred to, as well as the HER2, these are biomarkers that actually can change over time. And as they change, the prognosis of patients changes according to whether or not that biomarker is present. And it actually, these biomarkers can change. And as a tumor becomes HER2 positive, that obviously provides the possibility for treatment. So as we're thinking about this, and I'll start with you, Dr. Erlichman, how can we address some of these issues in terms of, first of all, the cost of doing the biopsy? Should it be borne upon insurers? Should it be borne upon the patient? How do we deal with that?
Well, I think that's one of the biggest challenges that we have today, certainly in the solid tumor area. I think if you look at the practice of oncology in general, and you look at what hematologic malignancies does, it's standard of care to repeat bone marrow biopsies at a certain time for myeloma, leukemia, etc. And we need to get into that paradigm. The insurance companies cover those costs because they know a decision will be made about a treatment option at that time. So to some degree, this is already happening in the hematologic malignancies, and obviously Dr. Fonseca can speak better to that than I. And I think what needs to happen is we need to demonstrate value, and not just value, unfortunately, not just value to the patient, but value to the insurer, meaning we can now show them that treatment X, which is going to cost $10,000 a month, is not worth giving the patient, but treatment Y is worth giving the patient. And then there will be convergence, and the payments will come along. But I think that we're in that sort of gray zone right now, transition period. I don't know if Dr. Fonseca wants to comment further.
Well, thank you. I think you summarized it very well. In the context of our protocols, we have paid for those biopsies, and out of research funding for that. And the same is true for the self-pay protocol, self-paid by the patient. I think, because of what Dr. Erlichman was saying, I think the answer to that question is largely economic and of discomfort. Because people who deal with CLL, where you can get tumor cells from the blood, is not an issue. It's not even an issue for cost, because it's part of just drawing blood again. So it really just is more of a practical one, because in reality, you would want to have that information, ideally. But if you have to repeat a brain biopsy for a glioblastoma, that's very hard to do. So there's just the practicalities that would dictate how that goes along.
Okay. I have another question, and Dr. Fonseca, this is for you. You mentioned this incredible example recently published in Blood of the different clones that were identified in this patient with multiple myeloma, ultimately ending in plasma cell leukemia. How can we make sure that we identify those clones up front? And is there any evidence to suggest that ultimately these clones, even though they're obviously present at a very low level up front, can be targeted initially?
It's very hard, because your curve is almost asymptotic to that. Because you could say, well, I'm going to sequence 1,000 cells, single cell. You still don't know if there's 1 in 10,000 or 100,000 cells. So it becomes a little bit of phenomenology if you go down for too much. Your hope is that within those clones, they'll have differences, but they'll have similarities too, which I should have made the point a little more clear. They're not different cancer cells. They're all related. They're maybe cousins or brothers, sisters, but they have some common links. So your idea is, do you have that weakness? Because they'll have the commonality, that we can get them all right from the beginning, because you will never be able to get them all. And I think no matter how sophisticated the technology, there's always a question that the next cell is going to be slightly different. So that is one of the big challenges. And again, the idea and the hope would be that what you target is so strongly targeted, or works so well in combination with something else, be that chemotherapy or immunotherapy, or that you get 2 targets that you're going to be able to target this. Now, one of my hopes would be that we can predict what the next step is in that clonal evolution. The analogy I use for this is, if you look at, say, foxes, I'm using too many animals today, but, going after rabbits, you can predict that over time, over decades, rabbits will become faster, jump harder, or get camouflaged, something. But they probably won't get wings. So you can probably anticipate some of the steps that the rabbits are going to have through that evolutionary process. And we have an extensive data set, as was shown by Dr. Taylor. Maybe we can go with the drug that hits now, and potentially the one that predicts the next hit.
I think that's a really good point. I was recently reading an article, and as it comes to the idea and the era of targeted therapy, we oftentimes think of these different targets, such as the PI3 kinase pathway or the VEGF pathway, as sort of, as someone once said, a whack-a-mole. And that is, you sort of hit one target and then another one pops up. But I think actually potentially a better analogy would be chess. And that is that we actually not only need to be hitting the targets, but as you said, we need to be anticipating what targets will arise. We have a question up here, and that is, we'll try to address with the group, and that is, should all patients diagnosed with cancer have genomic testing? Dr. Erlichman.
I think Dr. Fonseca actually put it very well in one of his first slides. I think routine testing, genomic testing, is not where we are today. Will we be there in 20 years? Perhaps. I think that there are diseases and treatments that we have that are very effective, and doing the testing at that stage for that disease may be unnecessary and really not add value to the care proposition. I think that with time, eventually everyone might need it down the road. It goes back, though, to what Dr. Fonseca just said. If you can anticipate what the genomic changes will be and your strategy for treatment is different, then you may want to do it earlier in the disease. But I don't think we're at a stage where all patients should have genomic testing at this point in time.
Another question that came up here is, are some cancer types better suited for genomic testing than others? Clearly, Dr. Fonseca, you just mentioned the issue of doing multiple biopsies for a glioblastoma multiforme as opposed to a CLL. So clearly there are those issues. We're going to move on to the next question here. In your opinion, what type of genomic testing is best for cancer patients? Is it panel testing, exome sequencing, RNA-seq? And the follow-up question is, if multiple tests are reasonable, in what order should they be performed? Rafael.
That's a great question. I think this is one of the biggest challenges we have. We don't know how to organize this. I would argue there's value in copy number, and you can get those results right away. For instance, for ACGH, you showed an example where transcriptome was more helpful than whole genome. You could see how a minor glitch, maybe less tumor cells, would have given us a negative if we had only done the DNA on that one. So, of course, I'm speaking as an academician, not as a practical policy person. You want to do them all. The reality is, maybe we won't, maybe we will. Once these things come down in price, they're going to be cheaper than an MRI. They're going to be much cheaper than a PET scan. And as Dr. Erlichman alluded to, if you actually can avoid futile therapy, you can avoid a month of just empirical trial and error of a drug that you should know through genetics is going to be worthless, you might be saving another $10,000.
Any other comments? I have a follow-up question here. For leukemia and lymphoma, how do you isolate tumor cells from, quote, normal? And do you still use blood for germline testing?
Okay, I'll take a stab at that one. We actually do cell selection. That's one of the most expensive things. I think someone alluded to, I don't know if it was Dr. Taylor, that a big part of the expense is not the sequencing, but what you do before. So, we spend sometimes hundreds, if not thousands of dollars preparing the samples. So you have to do magnetic bead selection, purify the cells. In some diseases, you can use the blood as germline. There's always anecdotes where it's sort of confounded because there were enough tumor cells. But for instance, myeloma, you can usually compare that to peripheral B cells. If someone's really a stickler, well, they can use other sources of DNA. But for the most part, you can use it. You wouldn't use it usually for CLL. There's a lot of contaminating cells. So it sort of matters, what specific disease it is. But the short answer is yes, you have to select for that.
Okay. Dr. Taylor, I have a question for you, and that is, you spent quite a bit of time talking about the standardization of these different platforms. How close are we to getting different groups and different peoples to work together to provide standardized platforms and readouts so that we can start looking across studies and comparing and validating, as you mentioned?
I mean, I think that we're close in that the big, the big consortium projects are already doing it, right? So you see standardized analysis in places like ICGC, the International Cancer Genome Consortium. And I think the problem is for other groups to actually be aware of those large-scale best practices that are being developed and actually adopt them and iterate back. So for almost everything, like just for variant calling, the 1,000 Genomes provides an excellent set of best practices for doing that. Very few people actually use them. So I don't think the problem is so much developing the best practices as this tendency for every site to reinvent the practice themselves.
Okay. All right, we have another question here. Dr. Fonseca, you mentioned that 30 years from now we will be practicing medicine very differently. Why not 1 year, 5 years, 10 years?
It's a great question. Two reasons why I didn't say next year or next month. The first one is hematologists are always accused of being pathologic optimists, so that would be number one. The second one is that humans don't like change. I don't want to sound like a motivational speaker, but I would invite everyone to open yourselves to the possibility of what genomics can bring. I think we've been through many years of discussions about the risks. The risks need to be acknowledged. But we need to give equal balance to the possibilities and the potential of that. And I think it's going to take time. They say, no one likes change except a baby with a wet diaper. So we're going to see what happens.
That's a great line, by the way. Dr. Taylor, do you foresee a future where analysis workflows are published with the publications? And if so, how do you see this happening technically?
Well, I dream of that future, and I'm also incredibly optimistic. So I expect it will happen. We interact with a lot of journals on trying to make this happen. Certainly, open access journals get it. The idea that analysis methods should be open and transparent fits with their view of the world. Technically, but I think it really needs to be driven by the community, right? That's how journals usually work. If the community demands that a publication is held to a certain standard, then that's how change will happen. If when we review a paper, we say, the analysis workflow is not here, I cannot review this paper, things will start to change. Technically, I think we can't force any one way of doing it. What I think is we need to set a set of requirements that an analysis has to meet to be considered reproducible, and any approach that meets those requirements would be acceptable.
Okay, thank you. Well, we are actually coming to the end of this session. I would just like to thank each one of you and also thank the speakers for an excellent presentation. As you can see, this is just a wonderful area. We're living in a wonderful time of medicine, but clearly a lot of challenges, and the approaches that are being taken to address these challenges, we hope, are actually going to really bring new medicines and new strategies and therapies to our patients to the bedside. Thank you very much.
Thank you. Thank you. Thank you.